Welcome back, everyone. I'm going to run through the notes on semi-conservative replication. They're going to build on the structure notes we talked about in the last video, so go back and watch that one if you haven't seen it yet. The reason I'm writing is because there's a couple things here which might make a little more sense if you actually see me draw them out. That's why I'm not just showing you the answers. We're going to go through it together. So first of all, we're going to talk about why DNA replication is important. Remember, DNA replication is the process of copying a cell's DNA. So every time a cell divides, both resulting daughter cells need to have a copy of the original DNA. So the reason is every cell needs that DNA because it has the genetic information for that cell, has information for all the proteins and all the functions that that cell might need to perform. So every time cells divide, you need a copy. You can see that in this example down here. So if you start with one cell, this shows you everything that has to happen to make a copy of it. Mitosis is something that you might remember from the past. We'll study again in a little bit. But if you look at what we start with, one cell with two chromosomes, they're shown here in black and white. And then we end up with two cells. Each of those has two chromosomes. Well, if you count the total number of chromosomes, 1, 2, 3, 4, two copies of this one, two copies of this one, somewhere in the middle we had to copy them. That is DNA replication. So that's the process of copying a cell's DNA. It's called replication. Sometimes it's called synthesis. So really important because before we do any kind of cell division, we got to copy the DNA. Otherwise, you're going to have two cells with not enough DNA. Okay, so how does that actually happen? That's all this semi-conservative stuff up here. Why we talked about structure first is because the structure of DNA explains how it's replicated, how it's copied, especially the fact that it's double-stranded. So we'll see why that's so important here in a second. So before they had done all the science and done all the experiments to show how DNA was actually copied, there were different ideas as to how that might work. And those are the three models you can see here. On the left is the conservative method, which shows that when DNA is copied, the original strand is conserved. So remember, these are just ideas. Only one of these three is actually right. You probably already know which one if you read the title of the notes, but these are the ideas. And if you can understand why the other ones are wrong, that'll help you remember which one is right. So conservative method says that the original DNA is always preserved. So it's used to make a copy, but the original stands stay together. So that means the old DNA is shown in dark and the new DNA is shown in white. So however many times you copy your DNA, you still have the original piece you started with. That one was disproven. So that's not how it works. Semi-conservative, this is the way it actually works. But that shows that when DNA is copied, the two strands get split apart. So the original DNA gets split and then is filled in with new DNA on both sides. So if you copy it once, you've got two copies. Each one has one original strand and one new strand. If you copy it again, you simply split the strands. So you end up with another one that has original and new, and another one that's new. That's what you got from the other strand. Same thing over here. That is the one that actually works. So that's how DNA is actually copied. We'll see some examples of that in a minute. Another idea was the dispersive model, and that says that old and new DNA just gets randomly blended up together. So every new piece of DNA has a little bit of old, a little bit of new, all scrambled up. Not how it works. So for the rest of this, we're going to focus on semi-conservative because that's what actually happens. If you are curious as to how they figured that out, there are some really cool experiments that showed that. We're not going to go into them today, but if you search for the Messelson and Stahl experiments, that'll give you a little bit more information. So the important takeaway from that is remember which one is actually right. So experiments have shown that DNA is copied using yep, you got it, semi-conservative method. Woohoo! Okay, here's what it looks like. If we start with a piece of DNA like this, so we'll use this. So we'll make a little key over here. My solid lines are going to be original DNA, and then I will do dotted lines for new DNA. The basic way that this happens is to copy DNA. First, you take the two strands and then you split them apart. Remember, that's possible because of the structure, because you have hydrogen bonds in between the two strands, holding the two strands together. Those hydrogen bonds are easier to break than the covalent bonds that hold the backbone together. So you've got enzymes that will come in and split the strands apart. So there's one of our original strands. There's the other one of our original strands. Then we have other DNA that'll come in and fill in the missing gaps on the other strand with new DNA. So once we've pulled them apart, we take new DNA and we put it in like that. And I'll show you on the back of this paper that if we do that, we end up with two pieces of DNA that are identical to each other, but they're also identical to the one we started with, which is what we want. We want like a photocopy machine. We want to make an exact copy of what we started with, so both cells get a full copy of the DNA. If we went through another round of DNA replication, the same thing would happen. First, you take these two strands, split them apart, which gives us one original strand, one new strand. Same thing down here. You get one original strand, one new strand, and then you fill it in with new DNA, putting in the complementary bases, A with T, G with C. And so after two rounds of replication, we have four copies of the DNA we started with. And notice that one of them is half and half, half old, half new, half old, half new. And then, so two of those, and then two that are all new. That looks exactly like what's happening here. So that just shows you the process of split the strands, fill in the matching bases. This is the overall pattern. Now we're going to zoom in and take a look at the individual bases and just double check that if we do that, we want to make sure that we do get an exact copy of what we started with. Okay, here we are on the back of the paper, and now we're going to go through the two steps of semi-conservative replication and make sure that if we follow those steps of splitting the DNA apart and adding complementary bases, that we actually get two DNA strands that are identical to each other and the same as the one that we started with. So we can see the bases here in the middle. Remember, they're hydrogen bonds that hold them together, hold the two strands next to each other. The 5' and the 3' you see at either end are used to mark the ends of the DNA molecule and to show you that DNA is anti-parallel. So that just means the two strands go in opposite directions. So 5 to 3, 3 to 5. So they're next to each other, but opposite directions. So the first thing we do is we take the two original strands and we split them apart. To do that, you have to break all the hydrogen bonds that are in here between the bases. And remember, that doesn't happen by itself. You need an enzyme to do that. So I'll write one strand up here and one strand down here. So that would give us, just copying down the top strand, 5', A, T, T, G, C, C, A, T, 3'. And then writing the bottom strand down here, we would have 3', T, A, G, C, whoops. All right, let's start over. I think I goofballed that one up. So 3', and we have T, A, C, G, G, T, A, 5'. And I'll go ahead and draw arrows here just to remind you that those two strands are the originals. All we did was pull them apart. Next, we're going to add more bases to each strand, complementary bases, A with T, G with C. And remember, that's also something that doesn't happen by itself. You just have different enzymes that will do that with the two strands. So here we go. Let's just add in the matching bases. 5' shows we're going to go in the opposite direction here. So that would be A goes with T, T, G, G, C, A, T. Finish with 3'. And then down here, 5'. And then A, T, T, G, C, C, A, T. 3' to make sure that they are anti-parallel. And then now look at the two strands we have. So we've got AT, TA, TA, AT, TA, TA, GC, CG, CG, GC, CG, CG, AT, TA, AT, TA, 3, 5, 3, 5, 5, 3, 5, 3. These two strands are identical to each other, which is good because we wanted two copies of each other. And they're identical to the strand that we started with up here. You can go all the way down the sequence, and both of these are exact copies of the one we started with, which means this idea of semi-conservative replication works. All you got to do is split the strands, fill in the missing bases, and each strand acts as a template for filling in the missing strand next to it, which is pretty cool that that's just kind of built in to the structure of DNA. And if you want the little satisfaction here, double check. Yes, strand 1 is identical to the original. And bing, you betcha, so is strand 2. Last thing we'll throw in here is some practice with Chargaff's rules. Remember, Chargaff's rules just say that A pairs with T, C pairs with G. So you may see questions like this asking you about the percentage of a DNA molecule that is one base or another. So the first ones, they kind of get a little bit more difficult as we go on. So if we have the percentages for 3 of the bases, we're looking at thymine. We know thymine has to be the same as adenine. So that's just going to be 35%. Remember, your percentages always need to add up to 100%. So 35 and 35 would give us 70. 15 and 15 would be 30. Check. That gives us 100% there. Over here, we know guanine has to have the same percentage as cytosine. So that's 29%. You can do the math, and that works out. So that's good. These ones get a little bit more difficult, but not really. So T has to be the same as A, 10%. C has to be the same as G, 40%. 10 plus 10 plus 40 plus 40, hopefully still equals 100. Similar deal over here, C and G, so 43. And then A and T, 7%. Not bad. Why are we doing these? Are these even difficult at all? Well, the last ones are kind of tricky. Every year when we do these, sometimes people are like, oh, you forgot to give me one of the percentages. I can't do these problems. Well, believe in yourself because yes, you can. So check this out. We know if thymine is 12% of the DNA molecule, automatically adenine must also be 12%. Bing. So how do we get the rest? Well, if we know 12 and 12, we've accounted for a total of 24% of the molecule, which means there is 76% of the molecule remaining that we haven't figured out yet. That 76% has to be G and C, and we know every C has to pair up with a G and vice versa. So if you take that 76% and just divide it by 2, split it in half, that gives you 38% and 38%, which gets to 100. Same idea over here. Thymine has to be the same as adenine, 30%. Together, those two make 60% of the molecule. We have 40% left over. That 40% is C and G. They have to be even because they all always pair up with each other, which means 20 and 20. Great. We're good at this. Okay. If you have any questions, please let me know. And if not, I'll see you next time.